Hello everyone, I am Mohammed Hamama, and welcome to your ASCP preparation camp. In this camp, we will cover all the topics on the ASCP lecture list. Today, our focus takes us deep into the realm of amino acids, peptides, and clinically relevant peptide systems. We'll also be laying the groundwork for proteins, with a deep dive coming in the next video. By the end of this video, you will have a thorough understanding of this topic and be one step closer to passing your ASCP exam. But before we dive, I want to remind you to subscribe to our channel and activate notifications to get updates on our new videos. If you find our content helpful, please hit the like button and share this video with your friends. Have you ever wondered what makes up the human body? What are the tiny molecules that keep us alive and functioning? Well, today we will be exploring the world of amino acids, peptides, and proteins, the building blocks of life. Amino acids are the basic units that make up proteins and peptides. They are small molecules that contain carbon, hydrogen, oxygen, and nitrogen atoms. There are 20 different types of amino acids that can combine in various ways to form different proteins. These amino acids join together in a specific sequence to form long chains, which we call proteins. These proteins are essential for the structure and function of our cells and tissues. Peptides are short chains of amino acids that act as messengers within and between cells. They control functions like appetite, blood pressure, electrolyte balance, and processing of carbohydrates and minerals. Proteins. These workhorses of the cell play a crucial role in keeping our bodies functioning properly. Proteins are long chains of amino acids, and they have diverse functions within the cell. Proteins are like the building blocks of our cells and tissues. They act as the building materials for our body, helping to repair and maintain our cells. But that's not all. Proteins also act as biological catalysts, speeding up chemical reactions within the cell. They are also movers and shakers within the cell, helping to transport molecules and maintain the cell's structure. Proteins are like the organizers of the cell, making sure everything is in its proper place and functioning correctly. They also act as channels and pumps for ions, helping to regulate the flow of substances in and out of the cell. And that's not all. Proteins also act as transporters, moving important molecules into and out of the cell. Our immune system relies on proteins to defend against foreign invaders, like bacteria and viruses. Proteins also play a crucial role in signaling within and between cells, helping to coordinate different processes in the body. Our genes contain the instructions for making proteins, and we used to think that we only had around 20,000 different proteins in our bodies. But recent research has shown that the actual number of proteins is much higher. This is because sometimes the instructions from our genes called messenger RNA get mixed and matched, resulting in different protein combinations. Other times, errors during cell division or outside influences can change our genes themselves, resulting in new protein variations. This diversity of proteins is crucial for our bodies to function properly and adapt to different environments. So the next time you look at a cell, remember that it's the proteins inside that are the real workhorses, keeping our bodies running smoothly. Amino acids, the building blocks of life. These tiny molecules have played a crucial role in the evolution of our planet and continue to be essential for our existence. It is believed that amino acids were among the first organic molecules to form on Earth, even before life as we know it began. Out of hundreds of amino acids found in nature, only 20 are essential for building the protein chains in the human body. These 20 amino acids, along with many others, play a critical role in the human body's shape, how it works, and its overall health. But what exactly are amino acids and why are they so important? Let's dive into the world of biochemistry and find out. Amino acids are organic molecules with an amino group, NH2, and a carboxyl group, COOH. Most have a primary amine group, NH2, but some have secondary or tertiary structures. Almost all protein-building amino acids, except proline, have the amino group attached to the second carbon, also known as alpha amino acids. One interesting fact about amino acids is that they have a unique structure that creates mirror image versions, known as D or L configuration. Our bodies mostly use L amino acids, while D amino acids have limited functions. Now, let's talk about the diversity of amino acids. There are 20 common protein-forming amino acids, each with a unique side chain, or group, influencing its properties. Other non-protein amino acids like beta and gamma amino acids also play important roles. The charge and pH of amino acids also play a significant role in their function. Amino acids can have positive, negative, or neutral charges depending on the pH and their side chains. At neutral pH, around 7.4, they exist as zwitterions with both positive and negative charges. Amino acids can act as buffers, but their effect is limited near physiological pH, except for histidine. The side chain properties of amino acids also determine their size, hydrophobicity, water attraction, and overall function. Some side chains are polar, attracted to water, while others are nonpolar, water-repelling. Charged side chains can be acidic or basic depending on the group they contain. And cystine's unique side chain can form disulfide bonds with other cysteines. Most amino acids are water-soluble, but some are not. Nonpolar amino acids are hydrophobic and tend to cluster together in the interior of proteins. Polar and charged amino acids are hydrophilic and tend to be on the surface of proteins. Amino acid supply and transport. Now we know that amino acids are essential building blocks for proteins, but did you know that they also have other important functions in the body? Adults need about 46 to 56 grams of protein per day, and even more during growth or illness. But where do we get these amino acids from? We get amino acids from dietary protein, which is broken down by enzymes in the stomach and intestine. We also get them from the breakdown of our own proteins. There are 8 essential amino acids that our bodies cannot produce on their own and must be obtained from food. Animal sources like meat, milk, eggs, and fish are complete proteins containing all essential amino acids. However, plant proteins may be deficient in certain essential amino acids, so it is important to have a varied diet. In some cases, such as liver disease, the body is unable to convert certain amino acids to others, making them essential as well. Amino acid needs to increase in certain conditions. Children, pregnant and lactating women, and people recovering from injury or illness have a higher demand for protein to support growth and development. Severe protein deficiency can lead to health problems like kwashiorkor, which is characterized by muscle wasting and edema, or marasmus, which is a general wasting of the body. So how do we know if we are getting enough amino acids? Blood tests for albumin or prealbumin can help assess our protein intake. Prealbumin is a good marker for short-term protein intake because it has a shorter lifespan than albumin. Our body uses various transport systems within cells to manage amino acids for different purposes such as metabolism, storage, and recycling. Defects in these transport systems can cause problems like hyperornithinemia, hyperammonemia, homocitrullinuria syndrome, or cystinosis. These disorders are characterized by abnormal levels of certain amino acids in the blood or buildup of cysteine in cells. One of the most crucial transport systems in our body is found in nerve cells. These transport systems are responsible for packaging neurotransmitters like glycine and glutamate, which are essential for proper nerve function. But have you ever wondered how these transport systems are controlled? Well, the answer lies in our genes. Genes determine which transport systems are produced in different parts of the body. A specific gene family called SLC encodes proteins responsible for amino acid transport. Different SLC gene families code for transporters in various locations such as the brain, gut, and mitochondria. Proper function of these genes and transport systems is essential for maintaining amino acid balance and overall metabolism. Amino acid metabolism. Amino acids also serve as starting materials for hormones, signaling molecules, and energy production. During times of high energy demand or limited food intake, amino acids can be converted into fuel sources like glucose or ketone bodies. Some amino acids, such as glutamine and glutamate, help keep the Krebs cycle running smoothly, ensuring efficient energy production. Other amino acids, like leucine and isoleucine, can be broken down to create ketone bodies for energy. Excess nitrogen from amino acids is converted to urea in the liver and excreted in urine. This process helps maintain the body's acid-base balance. Urea, though considered waste, plays a crucial role in concentrating urine, allowing the body to conserve water. Amino acids also act as precursors for many hormones, such as thyroid hormone and adrenaline, and signaling molecules, like nitric oxide. These molecules help regulate various bodily functions. Some amino acids, like serine and glycine, participate in single carbon transfers, which are essential for the synthesis of purine and pyrimidine, the building blocks of DNA and RNA. Folate, a B vitamin, is crucial for these single carbon transfers and overall cell health. Deficiencies in folate can lead to birth defects, and folate-based drugs are used in cancer treatment. Did you know that the concentrations of these essential molecules, amino acids, in our blood can vary greatly? In fact, even in healthy adults, these levels can fluctuate by 30% throughout the day. While diet plays a role, other factors also influence amino acid levels besides what we eat. Concentrations tend to peak in the afternoon and hit their lowest point between midnight and 4 a.m. After eating protein, amino acid levels rise in the blood and then return to normal within a few hours. But to measure true fasting levels, we need to avoid food for an extended period. Most amino acids are filtered by the kidneys but then reabsorbed back into the bloodstream. Only excessive levels or specific transport system problems lead to increased amino acid excretion in urine. Interestingly, amino acid concentrations in the brain are much lower than in the blood. This suggests specialized systems actively transport them across the blood-brain barrier. However, the main excitatory neurotransmitter, glutamate, is an exception and has similar levels in both brain and blood. Traditionally, doctors checked amino acid levels to diagnose inborn errors of metabolism. But now, researchers are exploring using these levels to assess other conditions like vitamin deficiencies, immune function, and even liver health. Measuring specific amino acids may one day help predict conditions like diabetes or preeclampsia. And tracking certain molecules could even give insights into blood vessel health. But how do we measure amino acid concentrations in the blood? Scientists used a method called cation exchange chromatography, which involved separating the amino acids and then detecting them using special dyes or fluorescent tags. However, this process could take 2 to 3 hours and was prone to errors from other molecules interfering with the results. That's why mass spectrometry, MS, is becoming the preferred method for amino acid analysis. It can identify and quantify amino acids in a matter of minutes, with high accuracy and sensitivity. Now the next step is peptides. What are peptides? Peptides are the building blocks of life, playing a crucial role in everything from muscle growth to immune function. Peptides are like smaller cousins of proteins, made up of linked amino acids but with a shorter chain length. Typically, peptides have less than 50 amino acids and a molecular weight under 6,000 Daltons. A peptide bond, like an amide bond, connects two amino acids. It forms between the nitrogen atom of one amino acid and the carbon atom of another. There are rare cases where the connection happens between side chain parts of the amino acids instead of the main chain. During protein synthesis, these bonds form between amino acids, guided by a special RNA molecule in the ribosome. Scientists can also create peptides in a lab for research or medical purposes. Peptide bonds can be broken down in two ways: nonspecifically using strong acids, or specifically using enzymes called proteases that target specific bonds between certain amino acids. The electron sharing in the peptide bond keeps it rigid and flat, but flexibility in the peptide backbone comes from rotations around the bonds next to the alpha carbon. These rotations are called Ramachandran angles and are crucial for determining the overall 3D shape of a protein. Now, we will be discussing the clinically relevant peptide systems. And we will start with the pro-opiomelanocortin system and its role in the human body. The POMC gene, found on chromosome 2, is responsible for producing a giant protein with 241 amino acids. This protein is then processed into smaller peptides, with as many as 10 different active peptides depending on where it's processed. These smaller peptides have diverse functions, influencing blood sugar and electrolyte balance, body weight and appetite, skin pigmentation, and pain perception. Currently, doctors mainly focus on just one of these peptides, ACTH, which interacts with the adrenal gland. Measuring ACTH levels can help diagnose problems with the adrenal gland, such as Cushing's disease or Addison's disease. To measure ACTH levels, doctors use special tests called immunoassays. These tests aim for high accuracy by targeting both ends of the molecule to avoid picking up inactive fragments. However, this approach may also detect larger, inactive precursor molecules related to ACTH, potentially giving inaccurate results. Additionally, ACTH tests can vary between labs, making it even trickier to interpret the results. Moving on to another group of peptides, natriuretic peptides play a vital role in regulating blood pressure and fluid balance. There are three main natriuretic peptides, ANP, BNP, and CNP. These peptides are released in response to high blood pressure or fluid volume, and they work by dilating blood vessels and promoting the excretion of sodium and water. Doctors can measure levels of ANP and BNP to diagnose and monitor heart failure, as they are often elevated in this condition. In the world of medicine, there are many molecules that play crucial roles in our health. Next molecule is hepcidin. Originally discovered as an infection fighter, hepcidin's main job is now known to be managing iron. This key molecule is a mature form with 25 amino acids, derived from a larger precursor protein. It has a tightly folded structure with several internal disulfide bonds. Hepcidin interacts with ferroportin, a protein that transports iron out of cells. By binding to ferroportin, hepcidin essentially flags it for degradation, preventing iron release. Anemia of chronic disease is a condition that involves low red blood cell count despite having enough iron stores. Increased hepcidin production is a major culprit. Measuring hepcidin levels can help doctors distinguish between iron deficiency anemia and anemia of chronic disease. It might also be useful for conditions like hemochromatosis, iron overload, and chronic kidney disease anemia. However, developing accurate tests for hepcidin has been difficult. The small size and high similarity across species make it tricky to create good antibodies for detection. Current immunoassays might overestimate hepcidin levels, especially in kidney disease patients where shorter, inactive hepcidin fragments accumulate. Improvements in test accuracy and standardization are crucial to fully understand hepcidin's role in health and disease, potentially leading to better clinical applications. Now, let's talk about another important molecule in our body, angiotensin. Renin, a hormone produced by the kidneys, detects a drop in blood flow or pressure. It acts on a large protein called angiotensinogen, cleaving off a piece to form angiotensin I. An enzyme called ACE further breaks down angiotensin I into angiotensin II, the key player. Angiotensin II has several actions in the body, including contracting blood vessels and signaling the kidneys to reabsorb sodium, both of which can increase blood pressure. Drugs like ACE inhibitors block the conversion of angiotensin I to II, lowering blood pressure. While important for treatment, measuring renin and angiotensin levels isn't very common in diagnostics. However, with advancements in technology, we may soon be able to measure these molecules more accurately and use them in clinical settings to better understand and treat various health conditions. The world of molecules like hepcidin and angiotensin is constantly evolving, and with further research, we can unlock their full potential in improving our health. Endothelins, or ET peptides, are produced by the lining of our blood vessels, also known as the endothelium. They play a crucial role in regulating blood vessel function. There are three main types of ET, ET1, ET2, and ET3, each produced in different locations in our body. ET1 is mainly made by the endothelium, while ET2 is found in the intestines and kidneys, and ET3 is produced in the nervous system. ET1 is generated from a larger precursor molecule and comes in inactive, big ET forms before becoming the active 21 amino acid peptide. This molecule is a strong vasoconstrictor, meaning it narrows blood vessels. This helps regulate blood flow and blood pressure in our body. However, elevated levels of ET1 have been linked to conditions such as diabetic kidney disease and high blood pressure. In fact, increased ET levels after a heart attack might indicate a worse chance of survival. Currently, there are tests available to measure ET levels in the body. However, their reliability is still uncertain, making it unclear how useful they are for diagnosis. Now, let's talk about another important hormone, vasopressin, also known as ADH. This tiny hormone plays a big role in regulating our water balance. Vasopressin is made and stored in the pituitary gland and is released into the bloodstream when needed. It acts on the kidneys, promoting water reabsorption and reducing urine output. This helps maintain the body's water balance. However, due to its very low levels and short lifespan in the blood, routine measurement of vasopressin isn't practical for diagnosis. Problems arise when the body either doesn't produce enough vasopressin, known as central diabetes insipidus, or the kidneys can't respond to it, known as nephrogenic diabetes insipidus. This can lead to excessive urination and dehydration. On the other hand, inappropriate secretion of vasopressin, known as SIADH, can cause fluid overload in the body. This can be caused by head injuries, tumors, or certain medications. To treat these conditions, a synthetic analog of vasopressin, called desmopressin or DAV, is used. It works by stimulating the release of von Willebrand factor from endothelial cells, which helps with blood clotting. Glutathione, or GSH, is a tiny molecule that plays a crucial role in maintaining our overall health. It is found in almost every cell in our bodies at high concentrations, and smaller amounts circulate in our blood. But what exactly is glutathione and why is it so important? Let's find out. Glutathione is a powerful antioxidant that helps maintain the proper balance of other important molecules within our cells. It acts as a defense mechanism against harmful molecules called free radicals, which can damage our cells and lead to various health problems. But that's not all. Glutathione also plays a crucial role in removing toxins from our body. It converts these toxins into a form that can be easily excreted, keeping our body clean and healthy. In addition, glutathione helps transport certain molecules across cell membranes, ensuring that our cells receive the necessary nutrients and get rid of waste products. While measuring glutathione levels isn't a routine test, scientists have developed techniques to do so, mainly for research purposes. These techniques involve converting all forms of glutathione into one type for easier detection. By measuring glutathione levels, we can get a better understanding of how this molecule affects our health and how we can improve it. In fact, genetic disorders affecting glutathione production can be identified by measuring the buildup of certain byproducts in the body. Don't keep all this valuable information to yourself, share it with your friends who might find it interesting and beneficial. And if you have any questions or comments, feel free to leave them down below. We love hearing from our viewers and we will do our best to answer all your questions. And finally, don't forget to ask for our ASCP short notes to supplement your studying. These notes are a great resource to help you review and retain the information we cover in our videos.